Okie dokie.
We have chapter 26. It's Phylogeny and the Tree of Life, however you say that. And then I have a video for you here. It's going to be one of the crash course videos. It pretty much talks about taxonomy, which is a category of...
Taxonomy.
It's
the science of classifying living things. That sounds exciting. Today, we'll basically be learning the Dewey Decimal System of evolution. It's like filing. Must be on the edge of your seat. Okay, shut up. When it comes down to it, this science doesn't just categorize organisms. When you look a little deeper, you realize it's telling the story of all life on Earth, and it's a pretty good story. Every living thing on this planet is related to every other living thing. If you go far enough back, we all have a common ancestor, an organism that both you and I are descended from, or something that a starfish and a blue whale are both descended from, or even weirder, that an oak tree and a salmon are both descended from. That organism lived. It lived very long ago, but it was here. And I dig that. The trick of taxonomy is basically figuring out where all those branches of the evolutionary tree are, and finding some convenient labels to help us understand all of these remarkable interrelationships. Let's be clear, though, taxonomy isn't about describing life in all of its ridiculous detail. It's mostly about helping humans understand it, because it's way too complicated without structure. To get that structure, biologists use the taxonomic system to classify all the organisms on the Earth. It's sometimes called the phylogenetic tree, or the tree of life, and it illustrates the evolutionary relationships between all living species. So there are about 2 million known species. There could be anywhere from 5 million to 100 million species. Scientists really have no freaking idea. New species keep getting discovered all the time, and the more organisms we have to keep track of, the more complex the phylogenetic tree becomes. So there's not always a consensus about how to classify this stuff. There's a lot of gray area in the natural world. Actually, let me rephrase that. The natural world is one giant gray area. Sometimes it's just hard to know where to put a certain group of organisms, and eventually the group gets so big the classification system has to be messed with to make room for it. So the system isn't perfect, but it's good enough that we've been using it for around 250 years. What's that? Do you spell a biography coming on? Carl Linnaeus was a Swede, born in 1707, and early in his career as a botanist, he realized that the botanical nomenclature of 18th century Europe was, well, just crap. For instance, in his day, the formal name of a tomato plant was Solanum caule inerme herbaceophorus pinnatus incisus racinus simplicibus. Linnaeus actually said, "Once I shudder at the sight of most botanical names given by modern authorities." Not only did the sloppiness bother him, he saw a whole sugar scrum blowing in, because new plants were still being discovered in Europe, but that was nothing compared to the crazy stuff that was coming from the New World. Linnaeus saw that pretty soon naming conventions were just going to collapse under all these new things to name, and then what? So Linnaeus famously started off by naming himself. He came from a peasant family, and at that time surnames were just for rich people. So when Carl went to college, they asked him for his surname, and he just made one up, Linnaeus, after the linden trees that grew on his family's homestead. Linnaeus got a medical degree and became a professor at Uppsala University, where he devoted himself to the study of nomenclature. He had his students go to places and bring back specimens for him to study and categorize. The method he eventually adopted was based on morphology, or physical form and structure. This wasn't necessarily a new idea. Back then, people grouped organisms by analogous or homopathic traits, structures that appear similar but actually come from completely independent origins. By this definition, birds would be more closely related to butterflies than to reptiles, because both birds and butterflies can fly. But Linnaeus had a good mind for this stuff and turned out to have a real knack for choosing actual homologous traits for his classification system, traits that stem from a common evolutionary ancestor. Linnaeus, of course, didn't know jack about evolution. Darwin wouldn't come around for another 100+ years, but he intuited that some traits are more important than others. For instance, he was struck by the fact that reproductive apparatus seemed to be a good way of classifying plants. He also caused a bit of a scandal by classifying class Mammalia based on the female abilities to produce milk from their nipples, because apparently that was pretty racy stuff back then. In his lifetime, Linnaeus cataloged roughly 7,700 plants and 4,400 animals. He published his classifications in a catalog called Systema Naturae, which by the time he wrote its 12th edition was 2,300 pages long. In the meantime, Linnaeus actually adopted a personal motto: God created, Linnaeus organized. Although taxonomy has come a long way since Linnaeus, we still use a bunch of the conventions that he invented. For instance, we still arrange things into taxa, or groups of organisms, and we still use the same taxa as Linnaeus: kingdom, phylum, class, order, family, genus, and species. We also still use Linnaeus's convention of binomial nomenclature, using a unique two-part name for every species: genus and its species name, in Latin or sort of Latin-ish. This practice actually started back in the Middle Ages, when educated people were expected to know Latin. We know a lot less Latin now, but we know a lot more about evolution, which Linnaeus didn't. And we have technologies like genetic testing to classify relationships between organisms, and yet we still use Linnaeus's morphology-based system because genetic evidence generally agrees with classifications that are made based on structure and form. However, because there was a lot of life that Linnaeus had no idea about, we had to stick a new taxa above Linnaeus's kingdom. We call it domain, and it's as broad as you can get. So domains are bacteria, archaea, and eukarya. The bacteria and the archaea are prokaryotes, meaning that their genetic material goes commando, with no nucleus to enclose it, while the eukarya make up all of the life forms with a nucleus, and include pretty much all of the life that you think of as life, and quite a lot of the life that you don't think about at all. It might seem like, since all macroscopic life only gives one domain, it's kind of silly to give prokaryotes two. And for a long time we didn't divide them up into different domains. They hung out together in a single domain called Monera. But it later became clear that bacteria, which live pretty much everywhere on Earth, including inside of you, with deep in the Earth's crust, and archaea, which are even more hardy than bacteria, have distinct evolutionary histories. Archaea being more closely related to eukaryotes, and yes, thus me and you. They have totally different cell membranes, and the enzymes that they use to make RNA, their RNA polymerase, is much more like ours. Under the domain eukarya, which is by far the most interesting and even occasionally adorable domain, we have kingdoms Protista, fungi, plantae, and animalia. Now, scientists have settled on these four for now. But these are categories that are a human creation, but there are good reasons for that human creation. The unscientific truth is that we looked at life and divided it up based on what we saw. So we were like, well, protists are single-celled organisms, so they're very different from the rest of the domain. And plants get their energy from the sun. Fungi look and act very different from plants and animals, and we already know what animals are, so they have to get their own kingdom. And though scientists are sometimes loath to admit it, that system of just looking and dividing things up actually worked pretty well for us. Not perfectly, but pretty well. But there's a reason why this worked so well. Evolutionarily, there are actual categories. Each of these kingdoms is a huge branch in the tree of life. At each branch, an evolutionary change occurred that was so massively helpful that it spawned a vast diversity of descendants. Plants, or plantae, are the autotrophs of the domain eukarya. Autotrophs meaning that they can feed themselves. Photosynthesis, of course. They're cellulose-based cell walls and chloroplasts, giving them a distinct difference from all other multicellular life. There are two other sorts of trophs. There's the heterotrophs, which get their energy by eating other organisms, and the chemotrophs, which are weird and crazy and only show up in bacteria and archaea, and they get their energy from chemicals. Now, the kingdom Protista is weird because it contains both autotrophs and heterotrophs. Some protists can photosynthesize, while others eat living things. Protists are basically a bunch of weird eukaryotic single-celled organisms that may or may not be evolutionarily related to each other. Scientists are still trying to figure it out. Some are plant-like, like algae, some are more animal-like, like amoebas, and some are fungus-like, like slime molds. Protists are one of those gray areas I was telling you about, so don't be surprised if by the time you're teaching this to your biology students, there are more than 4 kingdoms in eukarya. Fungi, which are, the funguses. They include mushrooms and smuts and puffballs and truffles and molds and yeasts. And they're pretty cool because they have cell walls like plants, but instead of being made of cellulose, they're made of another carbohydrate called chitin, which is also what the beak of a giant squid is made of. Exoskeleton of a beetle. Because fungi are heterotrophs, like animals, they have these sort of digestive enzymes that break down their food and get reabsorbed. But they can't move, so they don't require a stomach for digestion. They just grow on top of whatever it is they're digesting. And and digest it right where it is. It's super convenient. And finally, we have Kingdom Animalia, which is the lovely kingdom that we find ourselves and 100% of adorable organisms in. Animals are multicellular, always. We are heterotrophic, so we spend a lot of time hunting down food because we can't make it ourselves. And almost all of us can move, at least during some stage of our life cycle. And most of us develop either 2 or 3 germ layers during embryonic development. Wait for it, unless you're a sponge. So like I said, we use this taxonomic system to describe the common ancestry and evolutionary history of an organism. Looking at the phylogenetic tree, you can tell that humans are more closely related to mice than we are to fish, and more closely related to fish than we are to fruit flies. So how about we pick an organism and follow it all the way through taxa, from kingdom to species, just to see how it works? I know. Let's pick this kitty, because I know she'd like it. Like that. So kitties have cells that have nuclei and membrane-surrounded organelles, and they're multicellular and heterotrophic and have 3 germ layers of cells when they're embryos, so they're in the kingdom Animalia. And they have a spinal cord running down their backs, protected by vertebra, and discs in between them, and they have a tail that doesn't have a butthole at the end of it, like a worm, which I'm really glad about. And that puts her in the phylum Chordata. Kitty clearly does not like this, so I'm going to put her down now. And the kitty lactates. It gives birth to young like a cow instead of laying eggs like a chicken. And they have fur and 3 special tiny bones in their ears that only mammals have. So they're in the class Mammalia. So she's more closely related to cow than to chicken. Good to know. And like a bunch of other placental mammals that eat meat like weasels, the mustelids, and dogs, the canines, kitties are in the order Carnivora. And they're in the cat family, Felidae, whose members have lithe bodies and roundish heads, and except for cheetahs, retractable claws. And they're littler than tigers and panthers, which puts them in the genus Felis. And then at the level of the species, the descriptions get pretty dang detailed. So let's just say that you know what a cat is. So the species name is Catus. And look at that. Felis catus.
No, Kitty, I could have that whole thing stitched onto a pillow for you to sleep on. You'll be cute.
Thank you for watching our taxidermy-ish, taxonomy episode of Crash Course Biology. We hope that you learned something. Thanks to everybody who helped put this episode together. If you have any questions for us, please leave them on Facebook or Twitter in the comments below. We will get to them, hopefully.
Okie dokie. So that was Crash Course on the Tree of Life and taxonomy. First off, to go over it again, the phylogeny is the evolutionary history of a species or group of related species. The discipline of systematics classifies organisms and determines the evolutionary relationship. Systematics uses fossils, molecular and genetic data to infer evolutionary relationships. Taxonomy is the order of division and naming of organisms. So the phylogeny, it encompasses the taxonomy. Just to be clear, they're not 2 separate things actually. That phylogeny is just taxonomy. Pretty much our taxonomy is phylogeny. Binomial nomenclature. Binomial is what it's called, I guess, by biologists for slang or something. It was instituted in the 18th century by Carolus Linnaeus to avoid problems with translations for biologists to refer to organism by a 2-part Latin scientific name. Linnaeus also grouped the evolutionary relationships in a branching phylogenetic tree, which is what we just saw. A phylogenetic tree represents a hypothesis about the evolutionary relationship. Over here is a phylogenetic tree, and it's just showing you what's related and where the species diverged, or the animal organisms diverged in their evolution. Here is another phylogenetic tree. This one is going to show you different things like branch points. Branch points represent the divergence of 2 species. So this point right here would be a branch point. This point right here would be a branch point, and so on. Sister taxa are groups that share immediate common ancestors. So like they have an example here, taxa B and C, they share a single ancestor, so they're sister taxa. Rooted are the branch points within the tree, often drawn farthest to the left, which would be where the ancestral lineage is, represents the most recent ancestor of all the taxa on the tree. So in this point, ancestral lineage would be our rooted branch point, or rooted point. Basal taxon refer to lineages that have diverged early in a group of history. So in this case, taxon G, they diverged super early compared to all the rest. It took a lot longer to get to the same divergence. And then we go into features. We have homologous features, which is, homology is a similarity due to shared ancestry. The more complex two similar structures are, the more likely it is that they are homologous. Analogy, whatever, as similarities due to convergent evolution. Analogous structures that arose independently are called homeoplasies, Greek for to mold in the same way. So just keep in mind that homologous share same ancestors, and analogous structures do not share the same ancestry. They're just like the flying squirrels in the sugar ladder would be a good example. Evaluating molecular homology, whatever. Systematics uses computer programs and mathematical tools when analyzing comparable DNA segments from different organisms. This accounts for deletions and insertions, mutations accumulating over time. This art here is showing you the first one, they're exactly the same. The second one, the G gets deleted and there's an insertion of GTA in the second one, in the second line of code or RNA. The third one's going to show you what happens when that happens. Oops. So we're missing the G, and then we have all the extra characters. And the fourth one, it shows you how the computer realizes, well, there's an extra GTA here put here, and there's a G missing from this spot. So this means that they're actually related, even though there's insertion and deletion errors throughout the generations. That's how it gets done. So if we didn't have that, a lot of times you're going to see a GTA and an extra G in that one, you'd be like, oh, they're not even related at all, because this one doesn't have it whatsoever, even though it's just errors. Okay, grouping within the trees are called clades. Cladistics is a groups organisms by the common descents. Again, clade is a group of species that includes an ancestral species and all of its descendants. There are three types. There's monophyletic, consists of the ancestor species and all of its descendants. So that would be A. We got the ancestor species, which would be right here, and then all of its descendants would be A, B, and C. Then we have paraphyletic, which consists of an ancestral species and some but not all of its descendants. So in this one we're missing the descendant G, so that's why we have all of them except for one and the ancestral. Then we have polyphyletic, which includes distant relative or related species, but does not include their most recent common ancestor, which is going to be C. We have a group from B and then all the rest. If we were going to have it where it was going to be monophyletic, then it would have to include all of this. So it would have all the ancestors and everything else. But it doesn't, so it's not. A shared ancestral character is a character that is originated in an ancestor of the taxa. A shared derived character is an evolutionary novelty unique to a particular clade. In this example, we're showing that vertebrates all have vertebrae. So that's a shared ancestral character. But the lancelet doesn't have that, even though we have a shared common ancestor, so they wouldn't have that. So to them, that would probably be a shared derived character. I'm not sure about that one. The hinged jaw, again, anybody that's derived off that is going to have a shared ancestral character of a hinged jaw. And again, when, let's say that the hair, that would be considered a, I guess it would be a shared derived character, is because it's something that's unique to these two instead of the rest of them. And then you got a Punnett square that shows, I think that's a Punnett square, that shows the likelihood of that stuff happening. Okay. In some trees, the length of the branches can reflect the number of genetic changes that have taken place in a particular DNA sequences in the lineage. So, this one just shows the changes. The length, or the length and stuff is not supposed to reflect time or anything. It's just supposed to reflect changes. But if you go to this one that actually lists the time and stuff, the branches are supposed to represent chronological time, and the branch points can be determined from the fossil records. So this one you would actually be able to cite, well, back in the beginning of the Centrozoic period, then you had the humans and the mice diverge or something like that. So it's just nice to know that for the top one you can't actually say anything other than the fact that they diverged. The bottom one you can say, well, that happened around this time. All of it's going to be speculation, of course, and a rough estimate, but at least you have some way of saying when and where. Okay, maximum parsimony, I'm terrible at pronouncing things, assumes that the tree that requires the fewest evolutionary events appearance of shared derived characters is most likely. So the top one is pretty much a square showing you the likelihood of there being a shared characteristic between a mushroom, human, and a tulip, which for some things, not very much, of course. And then the bottom one, you can see that one side makes sense and one doesn't. You're not going to say that you have 15% of the DNA is shared with a person and 15% is shared with a mushroom, compared to saying that 5% is shared with a mushroom compared to the 10% that's shared between a human and a flower. So it just doesn't make sense to say that there's more shared between a human and a mushroom when that's not even feasible. The principle of maximum likelihood states that a given certain rule about how DNA changes over time, a tree can be found that reflects the most likely sequence of evolutionary events. I cannot remember what that means. All right, phylogenetic brackets allow us to predict features of an ancestor from features of its descendants. Example would be dinosaurs. So they're showing a common ancestor of crocodiles, dinosaurs, and birds, and then it diverging to be, you got your horn-looking dinosaurs, and then you got your mean-looking dinosaurs, and then you got your birdies. And they're saying that, and your crocodiles. They're just showing that at that point, that's when the features would start changing, and then one would not carry on another feature that is dominant. Another, like the crocodiles are going to stay the same, and yet the other ones are going to still change. Or they could diverge into other things from their own thing, which is probably going to happen. Relationship reinforcement. So this is saying that birds and crocodiles share several features: four chambers heart, song, nest building, and brooding. These characteristics likely evolved in common ancestors and were shared by all of its descendants, including dinosaurs. The fossil records support this by finding fossilized nests and the brooding dinosaurs. So they can pretty much say that the remains of these nests had fossils with animals that have the similar features. In this case, they're saying that they have eggs, the front limbs are the same, and hind limbs. So they're being able to say that the dinosaur is pretty much laying on top of its nest, and that's how it died, and those features apparently are the same as any other relationship between crocodiles and birds. All right. An organism's evolutionary history is documented in its genome. There are two types of homologous genes. We have orthologous genes. Homology is a result of a speciation event and occurs between genes found in different species. So that's going to be the first picture here. Let's say that a dog needs not to be able to see, what is the example I had? Dog and a human both have the ability to smell something with the olfactory gene. So we both have one, but they're not exactly the same. Or no, this was the electron transport chain, wasn't it? Okay, so a dog and a human have the same gene for an electron transport chain that we use biologically. We both have to have it, so we both have the gene. They're just not exactly the same, but they are. They're just not in the same spot in our sequence or whatever. But we both have the gene, so we both are going to have the ability to use the electron transport chain. But the paralogous gene is a homology result from a gene duplication where multiple copies of that gene have diverged from one another into a species. So let's say the second one here is the gene for smelling. Humans have 27 different ones or something like that, I read, while a dog or a mouse, I think it was, had way more, like 3 or 4 times more. It's because this gene was duplicated, but I don't want to say with errors, but like they're just different, so they have different amounts of being able to smell. Like ours is not nearly as sensitive as theirs. So they have a lot more of these genes than we would ever have. And the reason they have that is because it's a paralogous gene, which means that it just duplicates but with divergence from one another. So some species of dog will have more than those in others, or so on and so forth. Oh, wait. DNA that codes for RNA changes at a relatively slow rate and is useful for investigating branching points hundreds of millions of years ago. So that's one way that they figure out how the phylogenetic tree branches. And then mtDNA evolves rapidly and can be used to explore recent evolutionary events, which I think I read that they can use that to help predict like how different things are going to evolve, because it evolves so rapidly that they can just keep doing it over and over and over again. A molecular clock. Molecular clocks uses a constant rate of evolution in some genes to estimate the absolute time of evolutionary change. In orthologous genes, nucleotide substitutions are proportional and to the time since they last shared a common ancestor. In paralogous genes, nucleotide substitutions are proportional to the time since the genes became duplicated. So what this is saying is that the time that it takes for an evolutionary event or a divergent in species, I'm guessing, or would be on a certain amount of, like, it would have a trend or There's a mathematical way of figuring it out. For mammals, it takes millions of years. You can see that sometimes it takes very short amounts of time, but then other times it's taking a longer amount of time. But no matter what, it's still on the slope in which where it's supposed to be so far. So we can predict things with this and also go back in time and try to explain things with a molecular clock. And then we have the domains. Like they said, there used to be 2 domains, and then they made it into 3 because of the differences between bacteria and archaea. Eukarya consist of all organisms that have cells containing a true nuclei. This includes many groups of single-cell organisms as well as multicellular plants, fungi, and animals. Bacteria contains most of the currently known prokaryotes, and archaea consists of a diverse group of prokaryotic organisms that inhabit a wide variety of environments. And then I have a Jeopardy game to go over all that. Sorry, I go really fast. All right. Let's start off. Go for it. All right, represents the divergence of 2 species on the phylogenetic tree. Yes. All right. You're up. All right. Is the evolutionary history of a species group or group of related species? It's the broader term. Phylogeny. Not that I can even say it. Phylogeny. Phylogeny. All right, go ahead. Try another one, guys.
We're the same one.
Analogous structures that arose independently are called Greek for to mold in the same way. Right? This goes along with analogous and homologous. The structures themselves are called homoplasties. What else do we want to try? Classifications 300. Classifies organisms and determines their evolutionary relationships by using fossils, molecular data, and genetic data. Close. Sorry, what? It's systematics. It's the system that uses it. All right. Go again.
It's all the genes that we have.
Homologous genes that are found in different species because of speciation. Uh, I don't know where it would be in the book because I've been spending so much time on the PowerPoint. I should have wrote hints for these.
Beginning of the letter and a word and the end of the word.
Is that the orthologist genes or whatever it is? The, yeah.
Boom. Nice. Yes. Ortholog. Go ahead and pick since you won.
What about this is my clad one.
Clade is a group of species that includes an ancestral species and all of its descendants. Which clade
Is
did
this
that?
like a true false? Or?
Nope, it's a type of clade.
Oh.
Is it ballistics?
What?
Ballistics.
What? That's not even right. No, it's supposed to be. I need my book. Yeah. Okay. We'll do this is my clade 200. All right, a clade that consists of an ancestor, ancestral species, and all of its descendants. They're gonna be the same.
And all of its descendants. It's the modified.
It is the same. And there, that one has the right one. All right, we'll do 300. Clade that consists of an ancestral species and some, but not all of its descendants. Para? Ooh. Where now? 400? I'll say you guys are getting that one. Clade that includes distant related species but does not include their most recent common ancestor. What did you say? Couldn't hear you.
Poly?
Yeah. Good job.
That divergence too.
Divergence 200. A group that shares an immediate common ancestor on a phylogenetic tree.
Is it sister taxa?
Good job.
Let's go divergence 3.
The branch point within the tree represents the most recent ancestor of all the taxa in the tree.
Is it rooted?
I'm sorry?
Rooted?
Yes.
Let's go 400.
A lineage that diverge early in the history of a group. Starts with a B.
Basal tax.
Good job. Next.
This is my planet path number.
Groups of organisms are groups of organisms by common descent using clades. Boom.
Are we the same 300?
Similarity in structures are due to the shared ancestry. Good job. A method for estimating the time required for a given amount of evolutionary change based on the observations that some regions of genomes appear to evolve at a constant rate. Why didn't I have that in the slide? This is the one that I showed you a graph of mammals and how fast they can evolve. And the consistency in which they do so. Good job.
Classifications too.
In the ordered division and naming of organisms is the ordering and division of and naming of organisms.
Is that taxonomy?
Yes. Tree showing the inferred evolutionary relationships among various biological species or other entities. What? Same thing. Phylogenetic tree. Is a group of one or more populations of an organism or organisms seen by taxonomists to form a unit. This was at the very beginning of this crash course video when Hank described them, the groupings. Taxa. Or taxon. Taxa is singular. A branch point from which more than two descendants taxa emerge and indicate that the evolutionary relationships among the descendants taxa are not yet clear.
Yeah.
Yes.
We would already say 200.
Similarity in structures are due to convergent evolution. What kind of structures are those? I'm sorry? Yep. Is a character that originated in an ancestor of the taxon. Ah. This is the one where I was talking about how vertebrates all shared the same characteristic.
Sister
A shared ancestral character. We'll do the 500. Is an evolutionary novelty unique to the particular clade? Because we had the shared ancestral characteristic, and then there was the other one.
shared derived character.
Good job. All right. A scientific discipline that uses nucleic acids or other molecules in different species to infer evolutionary relationships. So this is the one I was talking about how we have to use computers, or we do use computers. Molecular systematics. Okay. Homologous genes that are found in the same genome as a result of genetic duplication. This is the one that I was talking about, how we only have so many smell genes while others have more.
Paralogous genes.
Good job. This principle states that given certain rules about how DNA changes over time, a tree can be found that reflects the most likely sequence of evolutionary events. It's the principle of maximum likelihood. All right. This was instituted in the 18th century by Linnaeus to avoid problems with the translation. Biologists refer to organisms by a two-part Latin scientific name. Well, you guys got the daily double. And you're all winners. Questions? Discussions?
First of all, I gotta show a little appreciation.
Thank you.
Okay, of what we're looking at, right? So this is all student generated, student derived. Teaching instrument like the Jeopardy game is awesome, so that's highly encouraged to put that in there. It does help you to kind of stimulate your thought process. So let's talk a little bit about taxonomy. Why do we need this? Why do we need taxonomy? Why do scientists need taxonomy?
So we can classify organisms into different groups.
Okay, but what's the next step with that? Why would that be important? I mean, if I have a scientist in Australia, they're classifying a marsupial, what does that do for me?
Consistency?
Is that
Consistency?
consistency? But what is that consistency going to evolve into? What's going to help us understand what's going on over there? We know what he's saying, right? Since we kind of understand the language of taxonomy, we can kind of decipher what that animal looks like even without looking at it. If we know that it's in this kingdom, it's in this class, and family, and genus, we as scientists would know that those have certain characteristics getting all the way down to that point, and then the genus species would be the last key to actually what differentiates it from other organisms. See, so if you have a proper description, you don't have to actually see the organism. You get a picture in your mind of what it looks like. By going through the taxonomy, you can say, yeah, it's got 4 legs, it's got fur, it produces milk, blah, 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 right on the line, see. So then when it gets to the very end, that's when it is, the differences, the distinctions become more important. And we can classify them into groups. So instead of saying, well, it's a bug, now it's a marsupial, we know the difference because of the classifications. We don't actually have to see that marsupial to know that it's not a bug, because we know that classification. So when you look at the literature, you're going to see the whole lineage of those organisms. Clear down to orders, genus, species, so that you can see what it is. Because if you look at a bug under a dissection microscope, and we will as soon as they start coming out, you'll see that 2 flies will look almost exactly alike. By the naked eye, you cannot tell the difference. But if you look at the wing, there's one little partition in the wing that's a little bit different than the other one, and that's a different species. But we can take it all the way down to Muscadet, or Diptera, or a Diptera and a Muscadet, and make it so that you can see exactly what that fly is. And knowing the species, that tells us that little dip in the wing makes it that species. But up to that point, I can tell that if it's Muscadet, if it's Diptera, that means it is a fly. I know that for sure. So I know this taxonomy is really kind of tough to know, especially the words. It's hard, a lot of strange words. Phylogeny, all these different words are hard to understand what they are, but it's really scientists' way of putting things in a proper categories, proper classifications. And as the video said, up to this point, when Linnaeus was doing it, they just named them whatever they wanted to. It could be a Boyrosaurus when they found a dinosaur. You know, that's my dinosaur. I discovered it. I go, I'm going to name it after myself. They don't allow to do that. We redid these again back in the 60s. We came up with some more structure to it so that you can't name things after yourself. But even when Linnaeus was naming things after himself. So it's changed quite a bit. We've got a lot of things that we look at as far as helping us to understand the analogous traits, the homologous traits. How are these animals similar, but how are they still different? Where did they come from? And I got a kick out of somewhere up there, they said it was a theoretical the phylogenetic tree represents a theoretical representation of what think that this is true, but we don't know all the steps to get there. Right, so we think, okay, well, it all came from lobe-fishes, but what's the separation between us and the lobe-fish? Is there some others that we haven't discovered yet that may produce more branches at some other time? We don't know that. So you have to keep in mind that all this stuff is flexible, and as we find more fossils and more of the fossil record, it may change. It may change. So what questions do you have about this type of material? What stimulates your thought processes when you talk about these clades and how things are put together? So if we talk about humans, how do we know that we're not different than human that's in China? Are we different than Chinese? Are we different species? Are we the same? What makes us the same species? What makes us different? What would make us different if we had differences?
Now, people that are born with an extra set of chromosomes are still considered human, right?
Yep. So is it the genetics that we have that makes us what we are as far as humans? We have 46 chromosomes and they're all in the right sequence, straight down the line, compared to, let's say, a greater ape that's 96% genetically similar to us. Is that what makes us different? What makes us the same? How do we know? What a species, what's the difference between one species and another? So what do you think some things would be that makes us the same species? Why are we classified as the same species? Looking back to the last 3 chapters also, look back at some of that information.
Because we have opposable thumbs.
Opposable thumbs,
Isn't that one characteristic?
that's part of our traits, definitely. Like beetle, we also have the ability to speak, right? Animals do also, I just feel like we haven't figured that out yet, but that's my theory. But what else?
Three bones in the ear.
3 bones in the ear, right? That
Oh,
came
yeah.
out in this little thing, that's part of it. Reproduction, we can reproduce with each other, right? That's one of the big things right there.
Yeah.
If it was a situation like a donkey and a horse, where they couldn't reproduce and have viable offspring, that's definitely a big hint that we're not the same species, right? So those are some things that we can look at. That's why we do these taxonomies, so that we can see what the differences are in these species, and maybe even though that they have differences, they're still able to reproduce and be viable. So why did they come up with the domain? What is a domain? Different cell structures? That's the most all-inclusive, right? Domain. Well, life is the most inclusive. In order for us to do this, we're looking at life. Right? We're not looking at rocks. It's a different study. So
Tree of
life
rocks.
is the most inclusive. The domains we divide up into 3. What are the 3 domains?
Bacteria, eukarya, archaea.
Okay. And we are eukaryotes. What's that mean?
We have nucleus.
We have a nucleus. What's in the nucleus? So let's break down eukaryotes. What are the parts of eukaryotes? What are the different divisions? Protists, protista,
Fungi.
fungi,
Plant. Animalia.
what's the B, there's an A. Plantae, right, plants, and last one, animalia, right? Okay, so those are really broad. So what's the difference between a protist, a fungi, plantae, and animalia? What are the differences? And these are the ones we're gonna study the most, right? This is probably the most common that you're gonna see, unless you're gonna go into some really tricky, geeky stuff in biology.
The protists are single cell.
Plants produce their own energy. Food.
Strong energy, some of them.
Yep. Some need other things for energy.
Protists are chemotropes, autotropes, heterotrophs, right?
Mm-hmm.
They can do all things. It'd be kind of cool to put a couple of those in ourselves. We wouldn't have to do anything. We could live off chemicals, we could live off the sun, we could eat other things. I think that'd be awesome. Let's just do a little endosymbiosis with those, right? Heck with mitochondria, they're kind of wasted. So how about fungi? Why did the guy go buy mushrooms? Because he was a fungi.
Reproduce with spores.
Fungi are cool. They're
Creepy.
really cool.
They stand in one spot, they're big.
You can eat them on your pizza, or you can take them and take a trip. There's so much differences that you can do with fungi. You know, the hippies in the old days, I'm sure people are still doing it now, but they have certain fungi that have hallucinogenic properties. Or you can have a morel mushroom that grows out underneath your trees, underneath in your pasture or wherever, and you can eat it right out of the ground. Such a variety. But what do they do? What do fungi do?
Break that.
Break dancers?
Toadstool.
Old school? Everybody break down. So.
Are they the ones with no moving parts, or
What's that?
are they the ones with no moving parts?
No moving parts?
Is that
How do they reproduce? Anybody know?
Spores.
Spores. They can reproduce sexually and asexually. That's kind of cool, really. Isn't that kind of neat? What are some things that we use fungi for in our lives?
medicine?
Medicine? What ones?
Penicillin?
Penicillin?
Yeah.
Okay, yeah. I'll buy that. That's where it came from. What else? What else can we use fungi for?
Eat 'em.
So what is the yeast? Is that a fungi, a proteus, a bacterium? What is the yeast?
That's
Oh, that sounds
Good
good.
question.
All right, I want you to find out what we use fungi for, and then what a yeast is and what we use yeast for. What are these things we can use yeast for? Okay? How about plants? We're gonna do that the next hour, by the way, so we won't have to do that.
Same for, like this, scene, there's a guy that basically confiscates the mushrooms, and he takes them, dries them out, and they'll spread. So he basically molds them and then, bakes them, and they become hard, molded
chapter on fungi. Who's got that? Okay. When we do that, we'll go down to the fungi farm. Fungi farm south here. That's what he does. He raises fungi. We'll go out and talk to him. Another thing too that came up the other day. Anybody interested in virtual reality? Do you know anything about virtual reality machines? Okay. We have a program up at Quad Cities in ATEC that has virtual reality equipment. And there's a possibility we can go up there for a field trip for that also. So that's going to come up. But tell me about plants. What makes a plant a plant? Photosynthesis. All plants have photosynthesis.
Something about the cell wall.
Cell wall. Okay. I'll buy that. What's in the cell wall that makes it special?
Cellulose.
So what do you make cell walls out of? Paper mache.
Steel walls.
What cellulose? Sound familiar? And what else about plants? So they produce their own food, right? They're autotrophs. They have cell walls. It makes a difference between us and them. The only differences between us and them.
They don't have a mitochondria?
Trying to think of what it is.
Well, we got another chapter on that one too. So let's talk about animals. We'll go through all this stuff. I want to special out too. Animals. What's it is about animals? Animals. What makes an animal an animal? That's just kind of weird to think about. You know, we all know what an animal is, but what does it make it an animal? What are the characteristics of an animal?
It's sad because you think you know what an animal is, but then you ask, and it's like, I have no idea.
That little word assume,
Yeah.
you know, it's what they always say, don't use the word assume, because you assume that you know what things are, but you don't. It's just like I asked my class a little while ago, what is concrete, what is cement made of? Concrete mix. Cheeto. That's a Bobby Boucher answer. What's in there? What's in concrete? I know we're deep in science, but not from that biology. But what's in concrete?
Water?
Run down the rock. What kind of rocks? Limestone. Good. Yeah, we're on the track. What do they make up of buffalo? Well, they have that. And that big quarry up there, what do they do up there? Dig big holes and let people die?
That was recent.
That's what they did.
That just happened, 2 weeks ago.
What are they taking out?
Rock?
Limestone, right? So then, but you get this bag from Menards, right? Anybody mix concrete by yourselves? Nobody makes concrete?
Just the bags of.
You add water and you mix it and it's done.
You put it in the oven and bake it, mark with a T,
No oven, you just leave it out.
mix it up with a shovel. So you take this powder stuff, right? It's kind of gray. Throw it in there. Put some water in with it. What else? Throw some sand in it. Okay, because if you just put that powder in there, will it work?
It's too brittle.
It falls apart. It crumbles. So put some sand in it. What else?
They put it in the oil.
Put some rock.
Depends on if you want texture or not. Throw rocks in there.
You put some bigger rocks, put fibers in it, some people put fiberglass fibers in it to make it hold better. So you have to have kind of a matrix that fits everything together. So, I don't know how we got off that, but that's one of those things that you don't know, you think, oh yeah, I know what cement is, but you really don't think about it, right? If you don't know the components to it. So when we say what is an animal, yeah, we all know what an animal is, we know what a lion is, and a tiger, and a bear. But what makes those animals, like a lion, what makes those animals different from each other? What makes an animal different than a plant? That's kind of something that we just assume. All right, so I didn't know exactly how long this would take for the presentation, so we don't have a lot for today. What I would like you to do is you can either do it now or before you come back on next time, is find the characteristics of the Proteus, the fungi, plantae, and animalia, and find out where the differences between. So Proteus, fungi, plantae, and animalia.